Restriction enzymes are small enzymes that cut DNA, and they do this by recognizing very specific sequences within a DNA strand. So it might be a sequence of 4 to say 12 base pairs, and every time that restriction enzyme locates its specific cutting sequence, it will make a cut in the DNA. Next week in lab, we'll learn a little bit more about how they actually cut, but today we're just going to look at what they cut. So let's say we have a linear piece of DNA, and there are lots of different restriction enzymes. We'll use one called EcoR1. This just means that it's come from E. coli bacteria. And let's say that EcoR1 cuts 2 times along this sequence. So the way that this works is this enzyme will actually attach to the sequence and cut it as if it were a pair of molecular scissors. And it's going to cut it every time it sees that EcoR1 restriction site. So what we are left with are 3 fragments. Now these fragments might have a specific length, and in your lab you'll see what those lengths are. So let's say that the length between the end and this cut was 1500, maybe this is 5,000 base pairs, and maybe this is 1,000. So when they're cut, you'll end up with fragments of the same length. And these are measured in base pairs. Now a different restriction enzyme is going to cut at a different place. So let's say that we use our enzyme BAM H1, I think is what it's called. And let's say it cuts right in the middle of this same fragment. And maybe this piece is 3,500 base pairs long. And maybe this piece is, let's see, what would that be? 1,000, 1,500, 2,500, 7,500. So this would be 4,000 base pairs for that piece. So when it cuts, you'll end up with a piece that's 3,500 base pairs and 4,000 base pairs. You can also cut the same strand with multiple enzymes. And if you do that, let's say we also cut this one with BAM H1, and if we cut it with ECO as well, you'll get your ECO piece. You'll get 2 pieces from BAM H1 and your other ECO piece. So let's say this is 1,500, 1,000. Probably going to just say this is 2,000, which makes this one 3,000. That math is not probably going to add up with what we have below, but I think you get the picture there. So we can also have a circular piece of DNA. You find circular pieces of DNA in bacteria and archaea. So we usually are talking about bacteria. Bacteria and other prokaryotes have a single circular piece of DNA, and you can cut those as well. So let's say that there's an EcoR1 site here and here. And let's say this is a total of 8,000 base pairs all the way around. Maybe this is 5,000 base pairs and this is 3,000 base pairs. So if we cut this, we'll end up with 2 pieces. We'll end up with a 5,000 and a 3,000 base pair piece. Let's say we have another restriction enzyme called HIND3, and maybe that one only cuts this plasmid one time. When it cuts this plasmid, not plasmid, chromosome. A plasmid is something else. When it cuts this chromosome one time, all it's going to do is make a cut here. So it's going to open up and give you a linear 8,000 base pair piece. So if you have a linear sequence and you cut it one time, it's going to give you 2 pieces. But if you have a circular piece of DNA and you cut it one time, it's going to give you 1 piece. The second part of our lab is running fake gel electrophoresis simulations of these restriction enzyme fragments on a gel. So if we were to cut this linear sequence with all of these enzymes, both of these enzymes, we're going to get a series of fragments. We've got a fragment at 1500, 2000, 3000, and 1000. Now in gel electrophoresis, your DNA is going to run from or move from the positive end towards negative end. When we do this in person, you'll see that we actually attach an electrode to either side. So we load our DNA sample into this well. DNA is negatively charged, and this gel is like a really thick jello. And what it's going to do is pull the DNA through that gel, and it's going to run based on size. So we've already learned and observed several times that larger molecules move more slowly. So our largest piece, that 3000 base pair, is going to run to a certain spot. 2000 will run a little farther. 1000 and 1500. So you can see that these bands resolve or separate on this gel. So if we cut it with both EcoR1 and BAM-H1, we're going to get 4 bands. If we cut it with nothing, we're going to get a band up here at the top. All of our DNA is going to stay together because we haven't cut it. There's only 1 piece. Let's say we cut it with BAM-H1. If we cut it with BAM-H1, we're going to get 2 fragments. We'll get this one that's 3500, so a little higher. Whoops, I didn't draw this right. And another one that's, say, 4000. And he would run, say, somewhere in there. Most likely this would be resolved just a little bit farther. Now, let's say we had a circular piece of DNA. We could do the same thing. Let's say we cut this one with Hind 3, and we have 2 pieces. Maybe this big piece is around 3000, and this little piece is, let's say, 500. Now notice that we cut it 2 times, we only have 2 pieces, and that's because the first one opens it up linear, and the second one cuts off a fragment. So for the rest of this lab, you're going to look at how you cut and the fragments that you make different combinations of restriction enzymes. Then you're going to draw those fragments on a gel. And then the last thing you're going to do is you're going to look at a gel that has already been run, and you have to work backwards and see if you can draw a map of a circular or linear chromosome based on what you're seeing in that gel electrophoresis.